This
Um,
conference will now be recorded.
so my name is Jennifer Dorton. I'm the Deputy Director for the Southeast Coastal Ocean Observing Regional Association. I'd like to thank everybody for joining our webinar today. This is the second in a series of webinars that is being hosted by GCOOS, the Gulf of Mexico Coastal Ocean Observing System, SOCORA, and our ocean acidification partners, the Gulf Coastal Acidification Network and the Southeast Ocean and Coastal Acidification Network. So we are kicking off these whole webinar series is to highlight connections between the Marine Biodiversity Observing Network, or MBON, and our SOCAN, ocean acidific, not SOCAN, I'm sorry, our ocean acidification partners. Next slide, Emily. So today we're going to be talking about monitoring animal movement. Overall, within this series, our goal is to advance science in support of resource management and the blue economy by strengthening our collaborations across MBON and ocean acidification networks. So all of our speakers today do have an affiliation or do work with one of our MBON project teams or are part of our ocean acidification networks. I'm not going to read off all the objectives, but our overall, objectives really are just improving our understanding of the state as I'm sorry, folks, I'm getting tongue-tied today. Improving the understanding of the state of science, some of the gaps that we have, and some potentials for future research and monitoring in the Southeast and the Gulf of Mexico. We really want to integrate science across the marine biodiversity and ocean acidification realms. And then at the end of this entire series, we'd like to synthesize some of the lessons learned and develop a white paper. And this could really highlight new potential partnerships, collaborative research opportunities, monitoring needs, and how we more strongly connect the Southeast and the Gulf of Mexico regions. Next slide. Well, I've already hit the first one. I think I've actually hit all three of these. So I'm working through our opening pretty quickly. So I'll go ahead and go to the next slide, Emily. It didn't change. There we go. So today we're going to have three 12 to 15 minute presentations. We'll have one focused on OA and two focused on MBON. And then we'll have about 15 minutes at the end for questions and answers. Emily will unmute everyone, and folks, if you could raise your hand and we can then monitor. Chris Simonello is going to help monitor and facilitate our Q&A session. And so she can kind of keep it more streamlined if folks would raise their hand. But if you don't feel like raising your hand and you don't want to ask your question out loud, please use the chat function. We will also be monitoring the chat, and we can ask speakers any question that you put in there as well. So our speaker talking points today, as we've said, we're going to talk through the state of OA and MBON science, think through some information gaps, and also really highlight some benefits of communicating and collaborating across these OA and MBON networks, and highlight some of the challenges to collaboration. And hopefully, at the end within the QA, we think through how to collaborate better. Next slide.
I was muted. Sorry about that. Hi, everyone. My name is Emily Noakes, and I am SOCORA's Communications Engagement Specialist. I am honored to introduce our three speakers for today's webinar. For the ocean acidification portion of today's webinar, we will hear from Dr. Luke Thompson, a research scientist in the Ocean Chemistry and Ecosystems Division of the NOAA Atlantic Oceanographic and Meteorological Laboratory. Luke's work is focused on microbial ecology and bioinformatics. His current research involves the use of sediment trap collected eDNA to look at the composition of organisms at the surface and in the deep ocean. His talk today will focus on the current and projected basin-wide plankton responses to increasing temperature and acidification in the Gulf of Mexico. For the MBON portion of this webinar, we will hear from two speakers. First, we have Dr. Neil Hammerschlag, marine ecologist and shark researcher, and he is the president of Atlantic Shark Expeditions and the executive director of the Shark Research Foundation. Neil is an investigator with the U.S. IMBON and chairs the BioTrack and BioSound working groups. An innovative STEM educator, he mentors students with the Black Girls Dive Foundation. His talk today will focus on tracking biodiversity by networking biotelemetry and bioacoustics monitoring. The second speaker for the IMBON portion of this webinar is Dr. Jennifer Mixis-Olds, the director of the Center for Acoustics Research and Education and a research professor in the School of Marine Science and Ocean Engineering at the University of New Hampshire. Jennifer holds a research position in the UNH Center for Coastal and Ocean Mapping. She is the lead PI of a National Ocean Partnership Program project focused on the development of the Atlantic Deepwater Ecosystem Observatory Network. Her research interests include ocean soundscapes, animal behavior and communication, and environmental effects of human activities. Her talk today will focus on harvesting information from the Atlantic Deepwater Ecosystem Observatory Network. Now, I am going to turn this webinar over to Dr. Luke Thompson to begin the OA portion of our talk. So Luke, if you could please click the button on the bottom toolbar that says share my screen and show us your slides. I can see your slides.
Thank you.
They look great.
Okay. Yeah, thanks for the invitation. I'm excited to share our work on ocean acidification and temperature effects on plankton in the Gulf of Mexico. And shown here is the NOAA ship Ron Brown, which went out in the Gulf in 2021 on the GOMEC 4 cruise. And that's the data I'll be presenting today. And I want to acknowledge the Ocean Acidification Program for supporting this work, as well as the NOAA Omics program. And my home at NOAA is the Northern Gulf Institute and Mississippi State University. And I work at AOML in Miami. So in the kind of lead up to this work, the motivation is that, we know that ocean acidification and warming pose major threats to marine biodiversity, and yet the impacts on this diverse and ecologically important group of microbes, the plankton, remains unclear. And so we wanted to know, how do plankton communities respond to OA? Can we predict plankton responses both in other parts of the Gulf and in the future? And how do can we use these plankton responses as well to indicate where there might be ocean acidification occurring? And so the methods we used, as I mentioned, we went on GOMEC 4 in 2021, and we sampled seawater and sequenced the bacteria or prokaryote fraction, as well as the eukaryote fraction of the sample. And I'll talk a little bit more about the methods in a minute. And then we linked those plankton communities with different ocean acidification and other environmental variables. And then we developed linear regression models for the major microbial groups in the, especially in the photic zone in the surface waters. And that revealed different effects of ocean acidification variables, temperature, and other environmental variables. And I should say right off the bat that the main person leading this research has been Sean Anderson, and you'll see him in a few of the photos in a minute. So briefly, as many of you know, environmental DNA is a really hot technology, and it's being used in a variety of areas. You can detect everything from microbes to mammals, including fish, plankton, various types of animals as well. And so in this talk, I'm going to be focusing mainly on the microbial fraction, the bacteria and the protists, microeukaryotes. So, but there's lots of other things we can look at in these samples and other samples, and in my group, we do a lot of those different things. And so essentially in that seawater, there's different types of DNA, either from tissues or waste material, from intact cells or decaying cells, or even free DNA. There's also RNA as well. So we're focused on the DNA. And so what we typically do is filter that seawater. I'll show you that in a minute. And then we can apply different omics methods to that. Today I'm going to focus on this middle one, which is called metabarcoding. This is targeted sequencing looking at a specific region. And although you see fish and sharks and things there, we're going to be focusing more on the invisible part of the biota, which is the microbes. And so on GOMEC4, we got samples from 54 different sites at 3 different depths, so surface, chlorophyll maximum, and close to the bottom. And we got triplicate filters at each depth. And I'll show you a map showing some of that resolution in a minute. And so the whole workflow is, goes from sample collection. In this case, we use the CTD and collected seawater from those bottles, from those Niskin bottles on the CTD. We then filter that seawater in the lab on the ship using a peristaltic pump and a little filter called a Sterivex, which is shown it's that white filter there in the center of the slide. We've developed a QR coded barcode label that we stick to those filters. This makes it really easy and easy to track and to process those samples back in the lab using a QR code reader. And then we use a vortexer to agitate the samples, get the material off of the filter. That's that blue vortex genie shown there. And then we use there's different ways to do the extraction. We use a magnetic bead handling robot called a Kingfisher that's shown in the lower right there. And then from that extracted DNA, we perform PCR, which is also called library preparation. We send that off to a sequencing lab and they send us back data, which we then analyze using our in-house workflows. So here's a few more shots of Sean on the Ron Brown collecting samples from the CTD and filtering the seawater through those Sterivex filters. On the left is shown what the filter looks like with some biomass on there. You can see that yellow color, that's the plankton there. And then you got a close up there of the Kingfisher and Sean working in the lab here at AOML. And so what we did was we used 2 different barcode regions. So we're targeting both the bacteria and archaea with a region called the 16S rRNA. This is the small subunit of the ribosomal RNA that we're amplifying and sequencing. And then for the 18S, this is for protists. And also we do capture some metazoa, although I won't talk about that much today. We got a lot of jellyfish and things like that, some crustaceans. But I'll focus more on the smaller stuff, the dinoflagellates and diatoms and copepods and those kinds of things. And so we have both those 2 different groups, and then we run that data through our tourmaline amplicon sequence processing workflow, which uses QIIME2 and of some various different taxonomic databases and tools used, including R and Python and that sort of thing. So this is another version of the map, but this is showing the different lines that we sampled in different colors. So from Florida all the way around to Mexico, as shown on the left with the contour lines, and then on the right are those same contour lines but showing you the depth profile. So the idea here is to show you that we really have some nice depth coverage. This is a log scale on the right from the surface down to over 3,000 meters, and then those surface stations are just colored by the line numbers, and then the other stations, or other samples rather, are colored by the cluster. So what we did was we took all the data and we clustered them into 3 clusters, and these basically corresponded to the surface, chlorophyll max, and deep samples that we took. And so another way to look at that is if we group all of the samples by those 3 clusters. So cluster 1 is surface and coastal, cluster 2 is near the surface and epipelagic, and cluster 3 is the deep mesopelagic samples. So what we're seeing here are all of the 18S or protist data. We've got different groups of dinoflagellates in the surface, and those groups kind of shift as we go from the surface to the deep. So this is a really nice way to get a a full view of all of the diversity in this data in just one figure. This is the same kind of view, but looking at the bacterial or 16S data. We've got a lot of alpha proteobacteria, a lot of cyanobacteria, and as we would expect, the cyanobacteria, which are light harvesting organisms, those become almost indetectable in the deep, and they're taken over by other groups. Another way to look at the data is we have a between sample, which is also called beta diversity, and within sample diversity, which is called alpha diversity. So we can see very clearly that both in the bacteria and in the protists, those three clusters of samples from the surface down to the deep, they cluster with each other, and they're separated from the other two clusters. We see in some cases in the case of the bacteria, we see an increase in diversity with depth, whereas in the protist data, there's relatively constant, or in some cases, a slight decrease in the median diversity across those different samples. But of course, there's lots of extra information in those different transects that we can dig into if we want to. But one of the main takeaways that I wanted to bring is that we were able to put all this data into some general linear models and be able to build models of the different groups' responses in response to the environmental parameters and the environmental parameter space of those environments. So we were able to identify the models that were most predictive of these different groups using the main environmental parameters that were measured, including temperature, oxygen, silicate, ammonium, and then of course the really important ocean acidification parameters like pH and DIC. And I'll show in a minute kind of how the pH and DIC data were used. But if we put all those data into these general linear models, we're able to predict we're both able to identify which environmental variables are either negatively or positively correlated with each group. And then we're able to predict the relative abundance of those different groups in different parameter space. So shown here is the response of the bacteria on the left and the protists on the right in response to DIC, temperature, and pH. And so this can be used to infer well, a couple of different things. So one is it can be used to infer the Gulf-wide distributions at the present moment. So this is in this case 2021. The dots show where all the stations are that we got samples, but of course we're only sampling up finite points in space relative to an entire Gulf of Mexico. So to be able to say, what are the abundances of these things across the whole Gulf of Mexico, we can interpolate from the various other stations that were gathered, and also just interpolate using some matrix math and to be able to infer the abundance of these different groups. So we can estimate what the different plankton and different groups would be at these different regions. Another thing we can do is we can predict how these groups are expected to change in the future. So using a few different models from GFDL and some assumptions based on predicted temperature and carbon absorption by the ocean, we can predict, or there are model predictions of changes in temperature, DIC, and pH. So what we then did was we plugged in those numbers from these two different models and from 2050 and 2100 into our GLM, and we're able to predict the relative abundance of these different groups according to those models. So we would predict that, for example, Synechococcus will increase and Prochlorococcus will decrease among cyanobacteria, or that Dinophyceae and Syndyniales, these two different groups of dinoflagellates, would also be predicted to decrease under this increased temperature and increased ocean acidification environment. And so how do we define what is a more acidic environment, especially when we're wanting to develop indicators? So if we look at all of the stations together, We have DIC on the left here on this histogram and pH on the right. And so we defined high DIC as greater than 2100 based on the full distribution of data, and low pH is less than 8. And so if we group the samples in that way and say, what are the taxa that are most indicative of high DIC and low pH, we're able to see that this Ostriococcus, this eukaryotic algae, is both abundant and also highly indicative of this low pH, high DIC type of regime. And so this shows that we can develop indicators that if we were to go out and see that Ostriococcus was high in a given environment, we might suspect that that is indicating that there's high DIC or low pH in that environment, and potentially that's an indicator of ocean acidification. And so I think I'm out of time, but I summarize the results here. We were able to cluster the samples rather by depth-dependent diversity patterns, and predict model and then predict the distributions both within the Gulf and across future scenarios. And an indicator that popped up as especially important is this pico-eukaryote Ostriococcus as indicating of high OA. And so this really is a way to predict what might be happening in the future and gives us testable scenarios to look at as things start to change. And we want to continue this work on the upcoming ocean acidification cruises from NOAA, add additional sequencing to get a deeper understanding, as well as to add particulate organic matter analysis to look at particulates and carbon nitrogen ratios, and also to leverage BGC Argo data and particle backscattering data to integrate into these models. So with that, I'll just love to thank all the people that contributed. Sean did a lot of this work, as well as Leticia, who is the chief scientist, Catherine, who helped with data, and a number of other folks and funding agencies. So thank you so much.
All right, thank you, Dr. Thompson, for doing that awesome presentation. We will have plenty of time for questions at the end of this webinar. So go ahead and write down your questions for Dr. Thompson so we can discuss them at the end. Now I'd like to hand over the presentation to Dr. Neil Hammerschlag. And Neil, we can see your title slide and it looks great.
Great, thank you so much. So I'm just putting on my video. I don't know if you want that, but I'll put it on for you guys. So yeah, I'm excited to chat to you today, and I'm going to tell you a little bit about some of the work we're doing. I'm actually going to turn off my video so it doesn't slow down the presentation. Okay, great. All right. Okay. Do you see my next slide?
Not yet. Okay, now we see it. Looks great.
Okay. So today, I think it's pretty clear to most people that one of the biggest challenges of the century is to reverse the biodiversity crisis and declines. And I think it's one advantage is now we have a lot of different tools to understand and collect information on biodiversity, including eDNA, which you just heard about, underwater videos, catch data. But I think there's obviously this need to, improve the spatial and temporal scale of this monitoring, and to be able to go from just having the data to actually having the information. So in addition, there's all these different data sets that are out there, and how do we integrate them, these different data sets, so that we don't have to reinvent the wheel, and that we can we can benefit from others' work and map them on one another to get a better information and better and to help understand and interpret what different types of monitoring tools will tell you. So I'm going to today tell you about a couple efforts that I'm helping to lead. The first one is called BioTrack, which is an initiative focused on using animal telemetry data to help inform biodiversity monitoring. And the other one is BioSound Initiative, which is focused on using collecting sound information from the environment to better understand biodiversity. And Jennifer next is going to talk to you a bit about sound, so I'm not going to get as detailed into the BioSound project, but just to give you an idea of some of the goals that we're working on. In terms of BioTrack, the main goals of this work is to identify and characterize, try to understand and identify areas that multiple species are using, particularly threatened marine animals. And the idea is to use animal tracking, like satellite tracking and acoustic tracking, to figure out where animals are spending the most time, especially threatened species. Where do they spend the most time, and where do these areas overlap? So what is the most important areas for many, many species, these biodiversity hotspots, particularly of at-risk species? And then once we've identified those kind of shared common areas that are important among a variety of different threatened species, is to ask, do they overlap with marine sanctuaries or marine protected areas? What about with wind farms or areas that are being leased for energy exploration and energy use, like wind energy farms or other types of ocean exploration? And to figure out, are we doing a good job of protecting these hotspots of threatened species, or are they exposed to fishing and pollution? If we're going to put in a wind leasing block or do underwater exploration, mining, things like that, we obviously would like to avoid areas that are probably the most biodiverse, that are important for the most at-risk species. So how do we identify those areas to avoid them? And then we understand that these areas might change over time due to changes in a varying and changing climate, so from like global warming. So what we're trying to do is predict how these hotspots are going to change into the future, and how then may it impact these overlaps within areas that we're really interested in knowing if these hotspots are overlapping, like are they found within MPAs, or areas that are going to be wind farms, or areas that are being explored. Obviously, a big challenge in understanding this work comes from the fact that we can't just easily see ocean animals swimming around. They are hidden under the invisibility cloak of the ocean. And that's where, new advances in technology can really help out. And one of the major tools that we're going to be using is satellite tracking, where we attach transmitters to the body of an animal. In this case, there's a photo of that tiger shark with a satellite transmitter on its dorsal fin, and this will relate the location of this tiger shark as it swims around. The other tools that we'll be using is acoustic telemetry. And essentially what acoustic telemetry is a little transmitter, it's about the size of a AA battery, that we surgically implant into an animal. In this case, we're going to surgically implant it into a shark in the abdomen. And essentially that little transmitter emits a high frequency sonic ping. And that ping moves through the water, and the animals can't hear it, but we deploy on the ocean bottom special listening stations. They are recorders that listen for those pings from those tags. And those tags, each tag is a unique coded ping, so the recorders will log that location. So when a tagged fish swims around one of these recorders, it will record the date and time that fish was there, giving us presence and absence information. Unfortunately, many researchers use these same tags and these same listening stations, these recorders, and there are networks set up that we can share our data among different scientists that use the same tools. In particular, this project right now is going to focus on the Western North Atlantic, Caribbean Sea, and Gulf of Mexico, where there are scientists that are tracking thousands of animals with thousands of these transmitters, and as well as these underwater receiver stations. And there are several networks, tracking networks, set up to help share data, including the FACT network, ACT network, OTN, and ITAG network. And to date, we were able to receive contributions from 51 principal investigators, which included 3,509 unique different of the receiver or these underwater listening station locations, data from 69 different species, including 43 million acoustic detections of different animals. And I want to do a quick shout out to Tiago Couto, who is my former postdoc colleague, who has been really taking the lead in analyzing some of this acoustic tracking data. And I'd like to point out all of this is working together with different networks, the Animal Telemetry Network, FACT, Atlantic Cooperative Telemetry Network, and ITAG, and the Ocean Tracking Network. And it is really the goal. What's been successful in this effort is networking the networks. These networks are set up that within your network, other scientists can provide the individual tagger with information on where their tagged animal went. But what we've done is leverage the network to obtain pretty much contributions from those scientists to be able to share data not only across species and areas, but regions, to understand movement at a broader scale. And what we've been able to do is take those data and use some modeling, particularly looking at where these species occur and what environmental conditions they occur with, to generate information on probability of occurrence. And so in this image here on the left, it's looking at the probability of occurrence for the great hammerhead shark in our study regions. And the brighter the color, it tells you there's a higher probability of occurrence. And essentially, this is modeled data based on using the tracking data, and we can identify individual cells that are a resolution of 10 kilometers by 10 kilometers, where these animals occur and in what probability. And then what we can do is we can do this again for all the species that we track. So for example, when you take 69 species, you then generate data like you have on the right, which provides within a given cell, within a 10-kilometer cell, what is the number of species that we estimate to occur there in a given cell. And what we can do even from this data further to analyze it is to actually get at hotspots, is we can look within a region. So we can now take this data and say, if we go to a given region, we can now and separate and kind of weight the species data by region. As we can say, within each cell, what percentage of all the species that occur in a region occur within that cell. So now you can actually start looking at relatively important hotspots, the warmer colors versus colder spots, the colder colors within a region. So, the image on the right is zoomed into kind of the southeast region, including Florida and kind of off of the Carolinas. And what you can see is there are certain areas that peak in red, where that cell contains almost all of the biodiversity of the tracked species within that region. So this is really promising. The next steps is to really start integrating satellite tracking data, continue to now take these hotspots and overlap them with the marine protected areas, industry, human use areas, and then forecast what's going to happen under environmental change. So I'm going to quickly just get into what we're doing with Biosound. And Biosound, again, is the work that is taking information from the ocean sounds, essentially sounds that are coming from the environment, particularly biological sounds that are produced by fish or whales or shrimps and dolphins, trying to use that information to glean something about biodiversity and how the environment is changing. So the goal of BioSound Initiative is really to collect, to analyze soundscape, being information that is coming from the ocean, sound information, to support marine biodiversity monitoring. And really, part of that is just at the very root is trying to understand the value of this type of data and metrics that are commonly used to measure soundscape biodiversity as an indicator of biological and anthropogenic change over time. And our group has started with a focused effort that is really developing, and excited to share it with you. And this work is really being spearheaded by my colleague Elizabeth Ferguson, who has just been doing an amazing job kind of as beginning to analyze these data sets. But we have already been able to receive, people have contributed sound data from 8 locations across the nation, from a variety of different habitats. We have data from inside Biscayne Bay off of Florida to the May River in South Carolina, offshore areas like off of the Oregon coast and off of the Washington State, from the Hawaiian Islands, from the Arctic, from Gray's Reef, 8 sites that vary in habitat, estuary versus seagrass versus offshore versus reef from different areas. And what we've done is we've taken these sound files and we've begun to analyze them. And so things that we've done is to look at how the sound pressure levels vary over space and time and habitat. And then what we've done is we have begun to take existing metrics that are commonly used to measure the complexity of sound from the environment as a measure of biodiversity, traditionally used in terrestrial environments. So there are, we've used about 60 different acoustic biodiversity metrics traditionally applied in terrestrial environments and run them through all the different audio files from the 8 different locations. And we've begun to look to see how these different biodiversity metrics relate to one another, how they differ over space and time across our locations. And what we've also begun to do now is look to see how they relate to other types of information. So we have started to correlate these biodiversity metrics with metrics of ocean habitat biodiversity. In particular, we've been evaluating the Seascape package. Seascapes are a habitat classification based on a pelagic and nearshore habitat classification based on different ocean classes of water and plankton. And we've been correlating that with what we're learning from the sound to see how related they are or not. And so I'm not going to get into further details because we're really at the beginning stages. But what I want to say is that we do have a very large, ambitious workflow, even at this initial stage, that we are currently working through across our different 8 data sets that have been contributed from these different regions and habitats. We're looking to see how these different soundscapes differ in different biodiversity metrics over space and time, how they differ when there's, for example, boat traffic around. And we're also looking to see how they correlate with other types and metrics of habitat, complexity, and biodiversity. So that.
Hey, Neil, we're going to have to wrap it up. We're running into our other speaker's time.
All right. Well, I'm wrapped. Thank you.
Thanks.
Thank you, Neil, for that great presentation. And like I said earlier, we will have a brief Q&A at the end of everybody's talk. So write down your questions for Neil. And if we don't have time for questions, then feel free to just email them in and we'll get your questions answered. So now I'd like to give the talk over to Dr. Jennifer Mixis-Olds. So take us away. Your slides look great.
Excellent. Perfect. That's exactly what I need. Everybody can hear me?
Yes, you sound great.
Perfect. Okay, so thank you very much for having me. I'm excited to share with you today some work we've done from 2017 to about 2021 along the southeast coast. It was called the ADION Project, Atlantic Deepwater Ecosystem Observatory Network. And this was not something that I've done myself. This was an entire team of people. And what I really want to highlight, it was through the National Oceanographic Partnership Program that this project was funded. It was contracted through BOEM with NOAA and Office of Naval Research partners, and we have a whole slew of partners. Today, I'm really just going to talk about the acoustic measurements that we've been making as part of our project, although there are many other aspects of the larger ADION project. So starting from the very beginning, when we responded to the RFP for this project, the requirements were for long-term monitoring of the mid and South Atlantic outer continental shelf, which is an undersampled area of water, multi-year, year-round measurements, integrated system of multiple platforms. So we used fixed platforms in the form of an ocean lander, mobile platforms, our vessel that we service the landers on, and a sailboat taking array measurements, and remote sensing, satellite information for the greater region. We were tasked with priority measurements related to soundscapes. So Neil did a great job of explaining the value of soundscapes. Biology, we did actual marine mammal sightings and net tows, physical oceanography, water quality, meteorological or air quality, such as wind and waves and temperature, surface temperature, and human factors related mostly to shipping. The goal was for the system deployed for ADION to be relocatable and comparable to other national and international programs, and to make all of the data publicly available and accessible. So how did we do this? We ended up with 7 locations here pictured on the map on the right, from the northern site, Virginia Inner Canyon, VAC, all the way to Blake Plateau or Blake Escarpment, BLE in the southeast. All of these landers had passive acoustics, and the ones with active acoustic systems for looking at water column backscatter for fish and plankton are colored green. And so we only had echo sounder systems on 3 of our 7 lander locations. So this area is really interesting. The Gulf Stream is the dominant feature in this ADION region. And if we look at our 7 sites, 6 of them are either directly in or very close to the Gulf Stream. And that northern site up at Virginia is one that is a little bit outside, so it's in colder water to the west of the Gulf Stream. So keep that in mind, because if you just look at our sea surface temperatures from satellites, you can see here that the sea surface temperatures at Virginia had a much greater seasonal signal than the more southern Gulf Stream sites of Hatteras and Jacksonville, just because of their placement within the Gulf Stream. If we look at bottom temperatures from the lander, you can kind of see they do overlap throughout that 3-year period. There's not much of a seasonal pattern down at the bottom, and it kind of makes the rest of all of the sites uniform when we start talking about bottom characteristics. So here's our main workhorse. This is an ALTO lander, a long-term autonomous observatory lander. We usually deployed that off of the A-frame. This is a picture of it sitting on the back of the RV Neil Armstrong. And the reason we chose to go with a lander versus a mooring is because of the currents of the Gulf Stream. So on the left-hand side, here's the subsurface current profiles generated by HICOM, the oceanographic model. And so this was for one of our Charleston bump sites. And you can see that at the surface, the currents are much, much stronger than they would be at the bottom. And seeing we're interested in passive acoustics and fast-moving water creates noise in our recordings, we wanted to have our instruments down at the bottom where the current was slowest. So on this lander, under the yellow hats are hydrophones. So we had a 4-frequency tetrahedral volumetric design passive acoustic system. So this allowed us to get directional information of detected sources. On the bottom, you see a square and a round transducer plate. These are our transducers for the echo sounder system, and you can see this tube, white tube sort of laying in the middle. You can see it sort of peeking out on the left-hand side. That's all of the electronics and batteries for the active system. The hard hats contain the passive acoustic system. They're AMARs from JASCO Applied Sciences. And then the flotation here, we've got two yellow or orange flotation hats. Also on these landers are an acoustic telemetry receiver, CTD, so salinity, temperature, and dissolved oxygen. We have a beacon, satellite beacon for tracking it when it comes to the surface, and redundant acoustic releases. So we were very lucky in this project with a community of help to be able to get a 96% data retrieval rate. We had landers deployed from 2017 to 2020. And as everybody knows for field work, you just never know when things are going to go wrong. We had one lander that released here in the bottom center. You can see its release cord got stuck, and we had to have ROV Jason on the right-hand side help us retrieve that. Looking at the center top figure, this is another lander at another site, our Wilmington site. You can see it's pretty much buried in a sand wave. This is not what we anticipated. It released and didn't come to the surface. Luckily, ROV Jason was on that cruise, and we were able to get this footage to understand why it didn't come up. And Jason just rocked it back and forth, and it popped up to the surface. And then we've got this lander on the left-hand side. This one, for some reason, surfaced prematurely. And we actually had the RV Alusha was down in Miami at the time and was able to track this down for us. So good karma and community involvement, because anyone who does oceanographic fieldwork offshore knows it's a team sport and a community. And so we're very thankful for everybody that was part of our community on this project. So moving into the passive acoustic data set, we ran a bunch of tonal detectors and click detectors, which were able to allow us to detect approximately 15 different cetacean species. And of this, we had a 0.5% manual validation because we had a lot of data. And so instead really of going into the methods, I really just wanted to talk about the results today, starting with the marine mammal call diversity. And this is just for Odontocetes. And we had visual sighting data collected during the field cruises that helped us identify the more common species. But as many know, visual surveys are not as easy as picking up the more cryptic species at the surface. So acoustics is definitely a good tool for that. And so one of the things that you can clearly see as you look across all of the different sites colored here on the left, from Virginia in red to Blake Escarpment in sort of the dark blue, we saw dolphins of every season at every site. We have very few killer whales. Blanesville's beaked whales were had high site fidelity at our lowest, our more southern site, Blake Escarpment here. It was amazing to see this site fidelity. It was unknown that these whales were out there at such a strong presence throughout the year, not a whole lot of seasonal presence there. Unlike the Gervais and Truesbeak whales, who had very high site fidelity at the Savannah site, which was a deep water site off of the coast of Georgia. So that was something that we learned from this data set. You could start to look at dots of sperm whales and different Kogia species and sort of kind of see some seasonal patterns, but we're looking into that more. Moving on to mysticetes. So these are your baleen whales. Here it's quite interesting. One of the things that we found most fascinating that was unknown is that minke whales were so seasonally present on the southeast outer continental shelf off of Jacksonville and Blake Escarpment. You can see the seasonal presence of fin whales, and you can see they don't go as far south as some of the other whales, especially the minke whales. So we can start to look at both annual and seasonal patterns of both mysticetes and odontocetes with this data. I do want to focus also on the fact that we're not just getting omnidirectional data. With this 4-channel tetrahedral design, we're able to get bearing to each of our detections. So this looks like a typical spectrogram. Here we've got time on your x-axis, you've got frequency on your y-axis. In a typical spectrogram, the colors would indicate amplitude or loudness. Here in this directogram, it indicates the direction from where that call came from over this approximately 4-minute stint here. So you can see up to 5 different minke whales vocalizing at the Savannah site pretty much over the same 5 minutes. So knowing which direction an animal came from and knowing that this guy here, from the southwest cannot possibly be the same animal coming from the northeast, we start to be able to get at absolute minimum numbers or density estimation just based on combining what we know of the direction that the call has come from, the bearing that the call has come from, how fast an animal can swim, and the propagation conditions. So over this 3-year data collection period, we can see the seasonal presence of minke whales at both the Savannah and the Blake site, using this directional information to infer relative abundance or minimal audible animals. So now I'm going to switch from passive acoustic to active acoustics. So with our echo sounder system, we get things, data that looks like this on the right-hand side. This is an echogram with time on your x-axis, depth on your y-axis. So the surface is here at the top because the instruments are at the bottom looking upwards. These are the 3 sites on the left-hand side that we had echo sounders, and you can see where we had good data is in green and where we had no data is in red. The red here for Virginia was because our lander got trawled early due to fishing, and then this Jacksonville data gap here was because that was one of the landers that got caught, and we redeployed a lander but didn't have another echo sounder system to attach to it, so we lost data this last year at Jacksonville. So I mentioned that we have multiple frequencies on these echo sounders. So what does that actually mean? It means that by looking at the difference in the echo strengths at the different frequencies, we're able to tease out different species or animal groups. So this was done by size class. It's called a dB differencing technique, and you need more than 2 or 3 frequencies to be able to do this. And because we did net tows, every time we went out to service the landers, this was done by Joe Warren at Stony Brook University, we were able to match the species that we were seeing in these size categories. So small was less than 20 millimeters. We got medium, 10 to 40 millimeters. Large, greater than 40 millimeters. These are non-resident scatterers. That's what NRS stands for. So you've got your copepods, your pteropods, krill, and larger krill here at the large ones. And we got some big copepods and chaetognaths sort of in your medium. And then you've got your resident scatterers. These are swim-bladdered fish or small resident scatterers such as siphonophores. And so they kind of group them into these size classes. And then we were able to look at the acoustics with those size class distinctions. So we were able then to match here on the top, this is from Virginia. This is just looking at the backscatter strength, the NASS, nautical area scattering strength, with time on your x-axis and strength of that volume backscatter on your y-axis. And so you can see over these, this is about a year of data, the seasonal pattern of stuff in the water. We're just going to call it stuff because this is volume backscatter. It's just the measure of the echoes in the water coming back from the stuff in the water column. With our multiple frequencies and our net tows for validation, we're then able to look at how those different scattering groups, our medium non-resident scatterers in blue, large non-resident scatterers in orange, and then we've got small in gray, and then we've got our resident scatterer large. These are our fish. You can see throughout the year how the different community structure or scattering groups change. And we've done that for all 3. I'm just going to show you one more, which is Jacksonville. where we had a really, really strong bloom here, and these were resident scatterers, so most likely fish, and a really strong bloom of our smallest, our resident scatterers in gray. So with this information, we were able to look at community structure. I just sort of summarized it here. The Virginia site was dominated by large non-resident scatterers, with some medium non-resident scatterers and large resident scatterers. Hatteras site was dominated by large non-resident scatterers in winter and spring, and then the large resident scatterers in fall. So we're starting to see that lead lag time in predator prey relationships. And then the Jacksonville was dominated by large resident scatterers with some small resident scatterers. So that's sort of the summary of data that's available, which is what I wanted to end on, because I said at the very beginning, part of this project was to make all of our data available. Even though this project is over, it could still contribute very meaningfully to MBON missions and objectives, because all of our passive and our active data is available through NOAA's National Centers for Environmental Information, NCEI. I mentioned but didn't talk about the tag detections. Neil did a great job of talking about that from his project, but I did want to say that all of our tag detections are submitted to the Ocean Tracking Network, so they're accessible to everybody else. And we have a project data portal, which is showed here on the right. There's the website. So all of our data processing products are there. All of our satellite products are there. All of our modeling products are there. So if you actually wanted to go through, you can download the Excel CSV files of the presence and absence of every hour of data for every marine mammal species we had. So you don't have to do that yourself. We've already done it. If you trust what we've done, you can download it right from our data portal. So that data is accessible for years to come and will hopefully be a really great resource as environments change along the southeast outer continental shelf. And I'd just like to end with the fact that, again, this was a huge team effort. Large offshore oceanographic project required a lot of people, volunteers, and students. So I'd like to thank all of them. Okay, thank you. Let me stop
All right,
sharing.
thank you. Yeah, thank you, Jennifer. So really quick, I just wanted to make sure that everybody knew about our next webinar in this series, which is going to be discussing the Flower Garden Banks National Marine Sanctuary, specifically regarding collaborations to meet data needs and identification gaps. Our ocean acidification speaker will be Dr. Jingpin Hu with Texas A&M University. And our MBON speaker will be Dr. Marissa Natal with Flower Garden Banks National Marine Sanctuary. We will send out registration information in the next couple of weeks to everybody who signed up for this webinar, and we'll also be posting it on the GCUS and Socorro websites and social media platforms. So that wraps up today's webinar. If you guys have some time to stick around, maybe we could answer a couple of questions. But I think I put in the chat my email address. If you guys could just send me your questions, and I will get everybody connected with our speakers so that you can get those questions answered. And if it turns out we have a lot of questions, then I'll send out a list of all the questions and answers to all the attendees so that you guys can know what we talked about.
I'm
So
happy to answer any questions too.
Okay, perfect. I'm opening it up so that anybody can turn their camera or microphone on. I know Chris Simonello with GCUS was going to jump in and moderate the Q&A session. If you wanted to ask any questions.
Yeah, I know our speakers are, I'm not sure if you have time to stay on a little bit longer. I understand those have to leave, but Luke, Neil, Jennifer, those were great talks. I'm sure there's a lot of questions I have, and I know I have a lot of questions, and I'm not sure, do we have any folks who are staying on? Feel free to raise your hand or just go off mute and ask your question. That will be fine. And Jennifer is monitoring the chat box for some questions. While we're doing that, I guess this applies to all three of your talks. When you talk about the changes, what baseline information are you going on to look at community changes under a certain set of conditions versus what you're seeing now? Are there historical data sets that you're pulling together, or are you creating these baseline sets as you're taking some of the other environmental data sets? Specifically, I was thinking of that on your talk, Luke, with the phytoplankton communities.
Yeah, I mean, we don't have there is well, there are previous GOMEC cruises, right? This was the fourth one, so we can go back to the third, first, second, and first to get the OA data. We don't have the omics data, however, so we're partially limited that way, but we are hoping to continue sampling on future years, and so we can see changes into the future. There's a limit to how far back in the past we can go. That said, we do have other samples from other parts of the Gulf that we might be able to integrate into this model, but not taken in exactly the same way for the same purpose in the same locations. So bit of a challenge, but yeah, that's why it's always good to get samples if you can get them. But of course, there's costs and things associated with all that. So it's not always an easy decision.
And I can answer for Adion. We one of the motivations behind our project was that the outer continental shelf is really an undersampled area. So there's not a whole lot of baseline data from where we were. In some places, that was the first data that really came out with long-term measurements that can be used as a baseline as things like COVID hit. So we have a COVID paper out and looked at that. This was sponsored at a time when there was the possibility of opening up the outer continental shelf for possible energy use or energy development. And so these measurements were specifically targeted as baseline to have something to compare it to if and when environmental or human use changes in these areas.
Thanks. Neil, did you have any other thoughts or comments?
Yeah, we rely on all the data. I would say that's the I think that's the basis. You're only limited what you can do, and I think the more historical data it will improve what you can do, and not only your own but others. Yeah. So it's great to see like Adion putting up their data available and accessible for use.
Thanks. And Kim Yates, go ahead. I see you have your hand up.
Thanks. Great presentation, guys. I'm going to start this with, I'm not a biologist or an expert in telemetry for sure. I'm just wondering, how quickly would some of these top-level animals, predators, whales, sharks, respond to a shift in downchain, lower and mid-trophic level organisms that might be changing due to these plankton shifts caused by OA and thermal stress? And I guess what I'm getting at is, do we have the data to start building those kinds of up and down the trophic chain responses to OA and thermal stress? And that might be a question for all of you, actually.
Go ahead, Neil.
Go ahead. No, I was going to say, yeah, I think, it's kind of, I mean, everyone has to get their own data set in line, but these are definitely the kinds of questions that as a group, I think, particularly in MBON, that we can start answering that because we're already kind of organized to answer that. So as long as I think it's, the big question to do that is having it more than just a snapshot in time, I think. So I think if you have spatial and temporal variation in the environment, and you have spatial and temporal variation in the tracking data, you can do that, and certainly even potentially within the in the eDNA aspect, Luke, since you would be collecting a lot of animals in the same time scale and spatial scale.
Yeah, I mean, we want to, at least for some of our projects, maybe this one we hadn't planned it, but we could go back and sequence the fish and higher trophic level eDNA and look for correlations. We have done that in another project in the Gulf, and we see some interesting patterns, with associating and correlating plankton responses to those fish communities, or plankton communities with fish communities, and there are some interesting correlations, so that I think there's a lot there that may have, at very least, it has predictive ability, but it may also have, underlying functional trophic reality, behind it. So I think that's pretty cool.
Yeah, and I'll say too, from an ADON perspective, we have a nice data set in time, high resolution in time, and only 7 units in space. And so asking some of those lead lag times on predator prey or even biophysical interactions between the physics and the lowest trophic level, that's a challenge. It's hard because these animals naturally change or naturally track variability in their ecosystem. Animals are really good at doing that. And so understanding what variability is within the quote norm and when that variability goes outside of the norm and how animals react to that is a pressing question that hopefully this data that we're starting to take now can answer. You need to know the variability naturally in the environment before you can determine whether it's outside that variability window.
And I don't know about OA and pH in particular, because part of the thing is I don't think we really understand what some of those impacts are on the biology. But what I can say is, like, from the sharks that we've been tracking, we see really fast, rapid changes to marine heat waves, and we see longer term change to climate change. And so we do see those animals respond really, really quickly to the environment. And in fact, I would say, like, monitoring the upper trophic levels is kind of gives you a bit of an indicator of what's going on lower in the food web, because they integrate everything above it. So they're to some degree a canary in the coal mine. And so if it's sometimes you can actually, by tracking these animals in real time, if you see some changes in movement, you can maybe start asking questions like, what's causing that? And certainly we've seen changes in the timing and the extent of migration patterns based on ocean temperature variability. And we do know that, like, there's been changes in right whale movement tracking changes in copepod, which are moving in response to changes in temperature and phytoplankton. So I think these things can happen, relatively rapidly.
Well, thanks to all of you. This has been really, I'm excited for the conversations that will follow on this in the white paper development, because I see a lot of opportunities for collaboration here. Out of respect for everyone's time, Emily, Jennifer, do you have any closing remarks before we sign off?
Yeah, just want to say thank you again to all of our presenters. This was great and very informative. And also, I know that all these presentations or webinars are being recorded, so we will have those available for folks to come back and view later, so we can share them with anyone who you think might be interested in watching them at a later date. Thanks, everyone.
Thank you very much.
Thank you, everybody.
Thank you.